So this talk is essentially about COVID glycosylation and glycans. And we're going to be looking at virus glycobiology and the SARS-CoV-2 infectivity as well. But to kick off with, I'm going to start giving you an overview of glycobiology in general, what it's all about, where to find it, what it means, and so on. Let's see if I can. Okay. So there's a literature list, as I said, which will support all of the stuff that I'm going to talk about in this talk, and something which will link up nicely with what Steve has just been talking about is that if you're looking for information on glycobiology, I mentioned this in the last talk, there's a new form of the new edition of the Essentials in Glycobiology. If you want to find out anything about polysaccharides, as Steve's just been talking about, then go to this website, because you can get this free, and it includes all of the diagrams, illustrations, and so on. And that's quite valuable. So if you want to browse a little through this book, that'll be a strong backup to what Steve was talking about just now. So the outline of my talk is, first of all, I'm going to tell you about glycobiology. What are glycoconjugates, glycobiology in the glycome? Why is it important? What are glycopolymers, and why is glycan structure important as well? I'll then go on to the viruses, talk a little bit about influenza viruses and the glycobiology and the COVID-19. So to kick off with just a couple of words that you will come across if you're reading this literature. All of the molecules that contain carbohydrate chains linked to any kind of carrier, whether it's a protein or a lipid, or whether it's a polysaccharide, as Steve's just been talking about, these are termed glycoconjugates. If you want to study these carbohydrates in their many forms, this is termed glycobiology, as you've probably realized already. Anyway, the genes that code for the glycoconjugates in any molecule, cell, or tissue, this is known as the glycone. Now, glycoconjugates are include glycan structures, in particular sequences, which are made up of individual monosaccharides. This shows most of the monosaccharides which are common to the different glycoconjugates you're likely to come across. And there is a coding system, diagrammatic system, which uses symbols, circles, squares, triangles in different colors to represent the different sugars. What I'd like to point out here is the hexoses, glucose, galactose, mannose at the top with the blue, yellow, and green circles. The N-acetyl hexosamines, so this is N-acetyl glucosamine and galactosamine. These are widely spread, as you'll see in just a minute. Fucose up at the top right-hand side with the red triangle. This is also commonly found in many glycoproteins. Going down to the third line, the uronic acids, glucuronic and iduronic acids. These are common in the proteoglycans, glycosaminoglycans, as we'll see in a couple of minutes. And right down at the bottom in the middle, N-acetyl neuraminic acid or sialic acid with the purple triangle. So these are the main monosaccharides that we find in glycoconjugates. Steve also mentioned that the sialic acids are a little more complex than they would appear just for a single sugar. The reason for this is that sialic acids occur in a number of different forms or modifications in nature. So whichever animal you're working in, whichever cell you're working in, whichever tissue and so on, you'll find sialic acids of all sorts of different types. Now, it's an amino sugar, so it has an amino group. It can be either N-acetylated, as in human, or it can be N-glycolated. In practically all other organisms, we find the N-glycolar as well. Now, in addition to that, as you can see round this circle here, there are additions. We can get acetyl groups on different positions, 4, 7, 8, 9. We can get methyl groups, sulfate groups, and we can get phosphate groups, which is part of the biosynthetic pathway, as we'll see in just a second. So there is a huge range of structures which can be expressed in different glycoconjugates, which adds to the diversity of the glycans that we find in nature. So here is the metabolism of sialic acid. If we look over on the right-hand side, we can see starting with UDP N-acetyl glucosamine and going down the sequence, we can see that we go through several stages: N-acetyl manosamine, phosphorylated N-acetyl manosamine, the sialic acid 9-phosphate, which is then loses its phosphate to generate the sialic acid itself, and then the activation of sialic acid to a nucleotide sugar, CMP sialic acid, and this is what we need in order to transfer sialic acid onto glycoconjugates. And if you look at the little circle on the left-hand side, you can see this pathway, which I've just been through, but we can see that in relevance to where it actually occurs in the cell. Most of the biosynthetic pathway is in the cytoplasm, but for unknown reasons, the activation of sialic acid to form the donor CMP sialic acid occurs in the nucleus, and no one has a good explanation why that should be at the moment. This CMP sialic acid is then transferred across to the Golgi system, which is where the transfer of sialic acid onto glycoconjugates occurs. And these will then go out into vesicles or into cell membranes and so on. We also have the catabolic system, where glycoconjugates from outside are taken up into the cell, or they're recycled from inside the cell through the lysosome. And the lysosome, whoops, I've gotten a bit too far. Let's go back. There we get the breakdown of glycoconjugates. Sialic acids are released, and then they feed back into the system that we've just been talking about. So there are a whole range of events in the metabolism of the sialic acids which are relevant for this important sugar. So let's have a look at some example of the different types of glycoconjugates that we can find. Right up at the top, we see the proteoglycans, and these are the macromolecules that contain the uronic acids: heparan sulfate, chondroitin sulfate, dermatan sulfate, and hyaluronan. And these are very large molecules that can be anchored onto proteins, or as in hyaluronan, exist as a single polysaccharide structure. On the right-hand side, we can see glycoproteins. The glycoproteins can be secreted, and they can contain, or they can be membrane anchored. So we can have both types there. and I'll show you a couple of examples in the next slide in just a second. We can have N-glycans, we can have O-glycans attached to glycoproteins. On the other side, here we have the EGF receptor structure, which has a number of odd glycans which are linked through either glucose or fucose, and these are unusual structures. On the left of this is a so-called GPI anchored glycoprotein. GPI stands for glycosyl phosphatidyl inositol, and if you look at the bottom, you can see this unique structure, which has a couple of long chain fatty acids, which anchors these molecules into the membrane of whichever cell they're expressed in. The reason for this is believed that there are enzymes which will cleave this particular linkage and then release the GPI anchored glycoprotein at whichever sites is required. On the right-hand side, right over on the right-hand side, we can see glycolipids, the glycosphingolipids, and these again are anchored into the membrane. Inside the cell, we have glycoproteins which have just a single sugar, N-acetyl glucosamine, which is linked onto serines and threonines in these particular proteins. So we have a wide range of different glycoconjugates and different types of glycans attached to them. Here, just to give an example, again, I'm not going to go through all the different types again, but the sperm glycocalyx shows a whole range of different types of glycoconjugates, as we've seen in the previous cell line. And here there are some examples with the different sugar structures shown with the circles and squares and so on. So plenty of examples here just to illustrate this particular point. So to summarize, what are the structures that we find in glycoproteins? I'm talking about glycoproteins here, not other glycoconjugates. First, we have the N-linked glycans. So this is N-acetyl glucosamine linked to the peptide chain through asparagine. O-linked glycans, which is N-acetyl galactosamine linked to the serines and threonines. We have the single N-acetyl glucosamine linked to the serine groups in particular glycoproteins. We have the EGF receptor structures where we have glucose and fucose linked onto the protein. We have an unusual structure where mannose is linked to tryptophan through a carbon link, which this is very unusual. This has particular functions. Again, there's not time to go into all of that. And you'll find this in the essentials of glycobiology if you're interested to follow up. And the GPI anchor at the bottom, as I mentioned just now. Here are just a few examples to illustrate the diversity and variety that we find in these glycans. Now, these are N-glycans. These are linked to asparagine. You can see it at the bottom of these three structures here. We can have N-glycans that contain just mannose attached to the first two N-acetyl glucosamines in this chain, or we can have a complex type. We have mannose branches, and there are two arms here which are terminated in sialic acid. Or we can have a hybrid, a combination of the complex and the mannose rich types. And here, this is just again to illustrate, there are a whole range of structures which actually occur in nature. We can find them on the mature glycoproteins. All of these have been described. We can have some on the left, for example, that have got no sialic acids, but a couple of fucoses attached. We can have some that have got one sialic acid. We can have some that have 3 and 4 branches on these chains. So this is a wide range of structures which are possible through the biosynthetic mechanisms that form the N-glycan. Here are the O-glycans. So this is the N-acetyl galactosamine linked to serine or threonine down at the bottom. And we get 4 core structures, core 1 and core 2 on the left-hand side, so the disaccharide and the trisaccharide. And on the right-hand side, core 3 and core 4. If we add a sialic acid to this N-acetyl galactosamine, the structures can't be elongated. So you're stuck with this disaccharide containing sialic acid shown at the bottom there. And of course, for each of these cores over on the right-hand side, you can see examples of extended core structures. These are all structures that we find in nature, and in particular in the mucins. All of these structures have been demonstrated to occur in mucins. How are these these structures actually put together? They're put together in the rough endoplasmic reticulum, the Golgi system, and are ultimately secreted or localized into granules or membranes. And we can see here across the top the N-glycans and the processing which takes place to the mature molecule. The O-glycans, again, through this system shown in diagrammatic form here. The glycosaminoglycans, the proteoglycan structures, the GPI membrane anchor, and the glycosphingolipids over on the right-hand side. So we can see there's a diversity of structures which are all put together effectively through the same system to get the glycoconjugates that are made inside the cell. This is a complicated system, and I'm not going to go into detail about this. You'll find this in the Essentials of Glycobiology book. Essentially, if we're looking at the processing and maturation of an N-glycan, there is a block system of the sugars with the glycans, which I showed you just now, attached to the peptide backbone. Here, this happens up at the top here, and basically what happens is this structure which is initially formed is processed, goes through a number of different steps before we get down to either the oligomannose, the complex types, or whatever type that turns up. And this processing follows a particular sequence, which is the same in most eukaryotic cells. For the process for the O-glycans, the processing, in contrast to the N-glycans, the chains are formed by stepwise addition. There isn't a block transfer as we find in N-glycan biosynthesis. And here, as an example, here we can see the processing the extension of the core 1 to 4 structures can lead to a number of different systems in structures, including sialyl-lactosamine, sialyl-Lewis A and X, and the STA antigen. So to look at that in a little more detail, here there is our core 1 structure down at the bottom. This can be extended through glycosylation transfer to generate a chain which contains this trisaccharide, sialic acid, galactose, and N-acetyl-glucosamine, and then the chain will be extended out this way. This structure then is a branch point to form these 3 structures which I've shown at the top here. The SDA antigen, which is one of the most common glycan structures that we find in colorectal mucins. There are also sialyl-Lewis A and sialyl-Lewis X structures present, but they are present at lower levels in the normal colorectal cells. And these are the abbreviations for the glycosyl transferases, which are necessary to form these particular structures. Again, a lot of detail you can find in the essentials of glycobiology to support this. Now, if we have all of this diverse range of structures, why is that important? So why is glycobiology important? The number of potential glycan structures that can actually be formed is vastly greater than the equivalent that we can find for amino acids or nucleic acids. And it's the chemical structure of the monosaccharides, their anomoricity, alpha or beta forms, the size of their rings, the number of the potential linkages through the different hydroxyl groups. That is what creates this diversity. And as an example, a trisaccharide, 3 sugars from the repertoire of monosaccharides that we have, can form more than 38,000 different structures. In contrast, with the 20 amino acids that we have, a tripeptide, we will only get 27. And of course, for the nucleic acids, it's only 3. So the question then follows on from this. Why is it important that we have this diversity? What this diversity does is that it lends itself to the creation of a biological code, which has been termed the sugar code, or the 3rd alphabet of life. Such a code enables the creation of structures which have specific biological functions. And probably the best example that you'll get are for the blood group antigens. Again, that's another story. But then the diversity is quite plain in relation to the biological functions that are necessary. Following on from this, the array of different glycan structures that we find in glycoproteins, and in particular the mucins, is very large. However, we're still chasing after the biological function relationships that for the glycan structures that we've actually been that have been identified. So, in other words, it's important to consider each glycan structure that we identify if we are going to understand the relevance of their biological significance. The background to this is also that characteristic glycosylation occurs not only in the individual molecules themselves, but also in cells and in tissues. So there is specificity here which demands this sort of diversity. In addition, the changes that we know that occur in disease, and you saw it in the last talk that I gave, that there is diversity in these different diseases that we know. And they also relate to specific tissue sites. We get lung disease, we get gastrointestinal diseases, and so on. So this diversity lends itself to the range of structures which can be formed. The changes are related in the same way. So we have to identify the changes that occur, and we need to understand how they will affect biological function when those actually happen. Oh, I can't quite read this. What does it say there?
I can't hear us, can you?
Yeah, I can.
Yeah, we can
can't,
read it, Tony.
the little bar doesn't work. So the bottom line. Oh, right. The other thing, regulation of the host mucosal glycosylation occurs through interaction with the microflora, and this has been termed Glycan legislation. Again, that's another story, and you'll find that in the essentials of glycobiology as well. This is important because it's relevant to regulation of the structures which we know occur. So, so it comes down now to glycopolymers, large molecules that carry glycans. We know that they're in relation to biological function. There are plenty of polymers of different types which have biological function. What we know as far as the glycopolymers are concerned is that the main protective surfaces of the human body, the epithelial surfaces, are coated with secreted and membrane-bound polymeric glycoconjugates. And we saw some examples in the last talk about this. And biological protection against the external environment involves recognition of the host and the of the host structures as well. So, what if we're going to look at these glycopolymers? What are we looking for in relation to biological function? First of all, we want polymers with clustered glycan side chains. We saw this with this sperm glycocalyx. Glycopolymers are those polymers which act as targets for interaction between microorganisms, and here we're talking about the bacteria, viruses, and parasites. In addition, polymers with viscoelastic properties, and I think Gleb has mentioned this. The viscoelastic properties of these polymers give mechanical advantages, mechanical properties to the mucosal defensive barrier. So this is gel formation with the secreted mucins, for example, which are part of the protective layer on the surface of the epithelial cells. I'm going to talk about a couple of viral diseases now, but in general, there are a huge number of different diseases where pathology relates to glycobiology. And if you want to check this out, you can find this in the essentials. So let's get onto the viruses. There are a number of examples where viruses have used the glycobiology as part of their pathological mechanism. Those viruses which have been demonstrated to show this this approach include influenza, which I'll mention in just a minute, HIV-1, Ebola, MERS, SIV, and the COVID-19. All of these utilize glycobiology, in particular the sialic acids, as part of their pathological mechanisms. Here is an example of 3 viruses, and this is simply to point out that the viruses, different viruses, use different methods to enter the host cells. So they want to get inside the cells if they're going to cause infection. And you can see here the influenza virus has a particular mechanism for getting the virus inside the cell. If we look at the herpes simplex virus, again, we can see there is a characteristic route of entry with this virus. And for the HIV/AIDS virus, again, a different mechanism, which allows these viruses to actually enter the host cells. Let's focus first on the influenza virus. We know that the influenza viruses occur as ABC types. There is a hemagglutinin present on the surface of the virus, which
Tony,
binds.
these are the virus. This is influenza virus, not the bacteria. Yeah, you did before. Yeah.
Yeah,
So
influenza virus. Yes. First of all, the hemagglutinin binds onto the sialylated glycans on the surface of human mucosal cells. And then another surface protein, a neuraminidase or sialidase, releases the virus through cleaving the sialic acid and allows the virus to enter the mucosal cells. And in this respect, this type of sialic acid and the linkage to the cell surface is important for the pathology. So whether it's a 2,3, 2,6, 2,8,9, the susceptibility to disease relates to the sialic acid and its linkage. So here is an electron micrograph showing influenza viruses. You can see the circular shape, and you can see the surface proteins round the outside as well. Here is a schematic of an influenza virus particle. You can see the circular shape of the virus particle in brown here, and you can see all of the surface proteins on the surface, on the outside of the virus particle, and the single-stranded RNA inside. Here is another diagram scheme to show the interactions of the influenza virus, and if we're looking here at the surface of the respiratory tract, right down at the bottom, which contains and the inset shows that here is the mucosal surface. Here is a glycoconjugate which has a terminal sialic acid, and the influenza virus hemagglutinin binds to this particular structure. The sialidase, neuraminidase, will then cleave this off and allow the release of the influenza virus particle, which then is able to enter the cell. If we move on to the COVID-19, the binding of this virus to epithelial cells depends on the spike protein S1. The SARS-CoV-2 spike protein S1 is heavily glycosylated. It carries both N-glycans and O-glycans, and the glycan structure of these N and O glycans has been worked out essentially. So therefore, the strategy to understand COVID-19 infection depends on the looking at the glycan structures, the repertoire of these structures, and how this relates to their biological function. Here again, electron micrograph of a SARS-CoV-2 virus. You can see the circular shape of the virus, and you can see the proteins are on the outside. And here's another high resolution image for the same viruses. The body of the virus is stained in yellow, and you can see the surface proteins quite nicely staining in green. Here is another picture of schematic of the SARS-CoV-2 virus. You can see the shape of the virus, circular shape of the virus here. Some of the other proteins on the surface, the E, the S, and the M protein. What is important are the the spike protein trimers. These have a particular structure, and these are important for the binding to the human epithelial cells. This protein, the spike protein, is present as a trimer, and it's on the surface of the virus, as I've shown you just now, and it is covered by a shield of N-linked and O-linked glycans. Here's another schematic showing the structure of the spike protein. This is a trimer. You can see the receptor binding domain right at the top of the molecule here, and a stalk region. They've all got their particular names for these regions. What is important is that in brown, you can see there are glycans all the way along this spike protein. So the glycans are an important part of the structure of this spike protein. We know that the spike protein contains 22 N glycan sites and 25 O glycan sites. And relevant to the perhaps to the biological function, the O glycan structures, 16 of these are located very close to the N glycosides as well. So if you look at that in a little more detail, over here on the left, the Y represents an N glycoside, and the yellow square represents an O glycoside. And we can see that there is expression of these structures all the way along the spike protein backbone. So this is a significant biological function, structure which relates to function. In order for the SARS virus to enter the cell, there is a proteolytic cleavage, which is necessary if this entry is actually to take place. And this cleavage site has been identified, and it is the cleavage is carried out by the furin protease, which is found in man. We also know that there are O glycans close to this cleavage site, and these may play a role in protecting or releasing the cleavage site for furin to act. So here is the diagram. Here you can see the peptide sequence. This is the cleavage site. This 4 amino acid sequence is what is recognized by furin, and cleavage occurs then at this site. And there are 3 O glycan sites where O-glycans can be attached, and working out what the role of this protection or release, if these are cleaved off. This is part of the ongoing understanding that's necessary to understand biological significance. This is another diagrammatic, just to demonstrate the interactions that are necessary for the virus to bind to the surface of human cells and ultimately to enter. So here is the spike protein. We can see this on the left-hand side here, and here are the two receptors in the human cells. The ACE2 and the TMPRSS2 molecule. These are targets for the binding of the spike protein to anchor it onto the surface of the epithelial cells and to enable entry into the cells for infectivity. So to summarize, to look at the role of glycans, so there are several situations where we know that the spike protein can interact with glycoconjugates which are present in the host cell over on the right-hand side of this diagram. We know that the spike protein will bind to heparan sulfate. We know that the spike protein will bind to any glycoprotein which has, as for example here, an N-glycan attached. We know that spike proteins will bind to sialic acids, which are present in any kind of glycoconjugate which contains these sialic acids. And we know that there are lectins in the host cell which will bind to the spike protein, because as I just showed you, there are lots of different glycan structures on the surface of the spike protein. The blood groups are essentially irrelevant because they will be blocked by the antibodies that normally recognize the blood group substances. So this this event probably doesn't take place. Okay, so if we're finally, if we're going to test for SARS-CoV infection, we need to have the kits that are going to be able to detect it, and a clinic where we can use these kits to actually detect the plus or minus. And how these tests work is summarized in this slide here. There are two tests which test for viral material, either the nucleic acid or the antigen. Here we can see on the left-hand side the nucleic acid based test. Again, these are standard procedures which are well established now, or an antigen test which recognizes the surface antigens that are present in the virus. And again, this is well documented, and the kits and the cartridges to carry this out are well known. In addition, you can use the same cartridges to test a blood sample that can be taken from whichever patient. So this is a summary of how these tests actually work. So, of course, improvements in the testing kits and the screening methods for the general public is an ongoing feature of the research and is going on in a number of different countries. Okay, so that's the conclusion for this particular virus section. Glycobiology clearly is a fundamental feature of the infection process for viruses, including the COVID-19, the SARS-CoV-2. And as I just mentioned, research is still ongoing to reveal more fine detail and hopefully assist in the design of effective therapy.